the Swiss cheese in your lunch sandwich and the cordite explosives they used to fire artillery shells during World War I actually share the exact same microscopic engine.
Yeah, it's pretty wild to think about.
Right. Today we are doing a deep dive into a stack of sources on microbiology and bacterial metabolism. Basically, we want to figure out exactly how microbes make energy.
And, it is a subject that really forces you to look at the world on a completely different scale.
Totally. We're going to look at the historical battles fought over understanding these pathways, clear up some really persistent myths, and explore why this tiny invisible machinery is basically the scaffolding of human survival.
Absolutely. We're talking about how biological entities they extract energy from their environment just to stay alive from one microsecond to the next.
It's incredible. But to do that, I think we first need to kind of reset our vocabulary, right?
Yeah.
Because when you or I use the word fermentation in everyday life, we're usually talking about culinary stuff.
Right, like brewing a stout beer or culturing milk into yogurt.
Or pickling cabbage for sauerkraut, yeah.
Yeah.
But culinary definition focuses entirely on the final product, like the food we actually want to eat.
Exactly. But the biochemical definition in our sources is far more rigid. And understanding why it's so specific requires looking at this universal problem that literally every single cell on Earth faces.
Which is just finding a way to generate power.
Yeah, exactly.
So I want to start at the absolute beginning of that process. Like, what is the actual starting line for cellular energy?
Well, the primary starting line, pretty much across the board, is a process called glycolysis, or, more specifically, the Embden-Meyerhoff-Parnas pathway.
Okay, that is a mouthful.
It is. But whether you're looking at a bacterium in a puddle or like a human muscle cell, the process usually starts by taking a 6-carbon molecule of glucose, so just basic sugar, and physically breaking it in half.
Okay, I want to slow down here. Just because the sources throw around a lot of terminology about like energy currency and electron carriers, why does physically splitting a sugar molecule matter so much to the cell?
So it all comes down to the chemical bonds. The bonds holding that glucose molecule together actually contain energy.
Right.
So when the cell uses enzymes to snap that 6-carbon glucose into 2 3-carbon molecules, which are called pyruvate, by the way, it captures a small portion of that released energy.
And that gives you what, like a net profit of 2 ATP?
Exactly, 2 ATP. And ATP is the universal energy currency of the cell. The specific mechanism for this is called substrate-level phosphorylation.
Which means what exactly?
Basically, an enzyme directly grabs a phosphate group and just attaches it to an ADP molecule, which creates ATP.
I was actually thinking of this glycolysis step like a worker at a factory getting a tiny $2 advance on a paycheck.
I like that.
Right. You put in the initial work to break the glucose and you get 2 ATP back. It gets you through the morning. But the sources point out that this process also generates 2 molecules of something called NADH.
Yeah, and that is the crucial second half of the equation. To break those chemical bonds in glucose, the cell has to strip electrons away from the molecule. But electrons are highly reactive. They can't just float around freely in the cellular fluid. They have to be safely transported. So the cell uses a molecule called NAD+ as an electron carrier.
And when it picks them up, it becomes NADH.
Exactly. When NAD+ picks up those loose electrons, it turns into NADH.
It's essentially a delivery truck. You load the loose electrons into the back of the NAD+ truck, and it drives away as NADH.
That's a really apt analogy, yeah. But here is the catch, and this is what drives all of bacterial metabolism. A cell only possesses a limited fleet of these delivery trucks.
Oh, wow. Okay.
Right? If the cell can't find a loading dock to empty those electrons and convert the NADH back into empty NAD+ odd, the entire factory shuts down. Glycolysis stops.
So the cell can't even get its $2 ATT advance anymore, and it dies.
Precisely. It's game over.
So the divergence in how microbes survive the different pathways they take, that's entirely dictated by where they park those delivery trucks.
Yes. The first path is aerobic respiration, which is what human cells rely on. If oxygen is present in the environment, the cell takes that leftover pyruvate, runs it through the Krebs cycle, and stir off even more electrons.
And those electrons go where?
They're fed into this highly complex piece of cellular machinery called the electron transport chain, or the ETC. It's embedded right in the cell membrane.
Okay, right.
And the final catcher, sitting at the very end of that chain to accept the electrons, is oxygen. Because oxygen is so electronegative, it pulls the electrons down the chain incredibly effectively.
And the energy payout from this is massive,
It's
right?
huge. You go from that initial 2 ATP advancing glycolysis to a yield of 30 to 38 ATP per glucose molecule.
Wow. Okay, so I understood the aerobic part, but the sources mentioned a second path, which is anaerobic respiration. And this path also uses an electron transport chain. But if there's no oxygen at the end of the line to catch the electrons, how does the chain keep moving?
Well, bacteria are incredibly resourceful. Microbes like E. coli, when they find themselves in a place where oxygen levels have just plummeted, they can substitute oxygen with a different inorganic molecule.
Like what?
They might use nitrate or sulfate as the final electron acceptor at the end of the chain.
Interesting. Does it work as well?
Not quite. The electronegative pole isn't as strong as oxygen, so the overall ATP yield is slightly lower, but it still utilizes the full ETC machinery and produces vastly more energy than glycolysis alone.
Okay. Which leaves the third path, which is fermentation.
Right.
So if a bacterium is in an environment with zero oxygen and no alternative molecules like nitrate or sulfate are around, those delivery trucks basically stay full.
The factory's on the verge of shutting down.
Yeah, so the cell bypasses the electron transport chain entirely. It just takes the loaded NADH trucks and dumps the electrons directly back onto an organic molecule.
Exactly. Usually the pyruvate it just created in glycolysis. The sole purpose of that maneuver is to empty the truck, regenerate NADD plus DOI, and ensure glycolysis can keep churning out its tiny 2 ATP profit.
I mean, I'm trying to wrap my head around the evolutionary logic here. If aerobic respiration gives you up to 38 ATP and fermentation only gives you the 2 ATP, why would a bacterium ever resort to fermentation?
It sounds like a terrible deal.
It seems like a fundamentally terrible deal.
Well, it is a terrible deal regarding metabolic yield. But, metabolism isn't just about maximizing profit. It's often about immediate survival.
Right.
What fermentation lacks in efficiency, it makes up for in sheer speed and mechanical simplicity. It requires no specialized respiratory machinery in the membrane. It requires zero oxygen.
So it's basically a fallback plan.
Exactly. In a rapidly shifting environment, fermentation is a robust, rapid-fire fallback strategy.
Yeah.
The bacterium takes a massive cut in efficiency to ensure it doesn't just suffocate the moment the environment changes.
That makes a lot of sense. And if fermentation is a desperate fallback when oxygen vanishes, it makes sense why early scientists define the process purely by that lack of oxygen.
Yeah, that's where Louis Pasteur comes in. In the mid-19th century, he actually coined the phrase la vie sans air, meaning life without air, as his functional definition of fermentation.
Because he observed that when you seal off a vat from the surrounding air, fermentation begins.
Right, so defining it by the absence of oxygen was a perfectly logical conclusion based on the macroscopic observations they had in the 1860s.
Well, wait, if we go back to anaerobic respiration, the ones using nitrate, those bacteria are technically also operating without air. So how did the scientific community eventually reconcile a definition fermentation that suddenly included two completely different sets of metabolic machinery?
Well, the short answer is they couldn't. Pasteur's original dichotomy, aerobic versus fermentative, it completely broke down once microbiologists discovered anaerobic respiration.
Because suddenly you had organisms respiring and using complex electron transport chains without oxygen.
Exactly. The boundaries of the definition had to be redrawn to reflect the biochemistry. Today, a process is only defined as fermentation if it generates ATP solely through that initial substrate-level phosphorylation we discussed.
And if it uses an organic molecule as the final electron acceptor to empty those trucks.
You got it.
Well, having a clean, rigorous biochemical definition is great now, but getting to any baseline agreement at all seemed to involve a lot of aggressive public arguments in 19th century Europe.
Oh, it really did.
The struggle to understand what was actually causing fermentation reads like a scientific soap opera.
It basically was. The central conflict began in France in the 1850s. Louis Pasteur was actually approached by local beet sugar distillers who were facing a huge industrial crisis.
Their vats were spoiling, right?
Yeah, massive vats of fermented beet juice were spoiling. Instead of producing ethanol, the vats were turning highly acidic and sour, ruining the product. When Pasteur examined samples from the spoiled vats under his microscope, he didn't find the large, round yeast cells you typically associate with alcohol.
What did he find?
He found tiny, rod-shaped microorganisms.
And from that single observation, he makes a sweeping declaration that fermentation is inextricably linked to life. Like, it is a biological process that only happens inside living organisms.
Which immediately drew the fury of this prominent German chemist, Justus von Liebig.
Liebig, right.
Yeah, Liebig championed the idea that fermentation was a purely chemical chain reaction.
Yeah.
His theory was that decomposing organic matter possessed unstable molecules that basically vibrated, and these vibrations triggered a chemical cascade in the surrounding sugar.
So he aggressively insisted that living microbes were merely a byproduct of the decay, not the actual cause of it.
Exactly.
So how did Pasteur actually go about proving Liebig wrong? Because the sources dedicate a lot of time to Pasteur's swan neck flask experiments, but how did that specific setup settle a debate over, like, chemical vibrations?
The swan neck flasks were brilliantly designed to separate chemistry from biology. Pasteur took a nutrient broth that would readily ferment and spoil and placed it inside a glass flask. He then heated the neck of the flask and bent it into an S shape, a swan neck. He boiled the broth to kill anything inside it. Now, the open end of the flask allowed air and any supposed chemical vibrations in the air to freely enter.
Oh, I see. But any airborne microbes or dust particles would get trapped in the curve of the glass neck and could not reach the broth.
Exactly.
So the broth had full access to the air but remained completely sterile. It never fermented.
It remained pristine indefinitely. But the moment Pasteur tipped the flask so the broth touched the dust trapped in the neck, fermentation began rapidly.
Wow.
Yeah, it definitively proved that fermentation was caused by the introduction of external living microorganisms, not spontaneous chemical decay. Pasteur essentially won the debate of his era.
But the historical twist is that Pasteur only had half the puzzle solved. Because in 1897, another German chemist, Edward Buchner, decides to run an experiment. He takes yeast cells, grinds them into a paste using sand, places them in a hydraulic press, and just squeezes out the internal fluid.
Sounds messy.
Very. And then he filters that fluid to ensure absolutely no living cells remain intact.
And Buchner actually intended this as a control mechanism for a totally different experiment.
Yeah.
He fully expected that this dead cell-free juice would be totally inert.
But instead, he adds the cell-free liquid to a sugar solution, and the liquid ferments the sugar into alcohol.
Yep. He accidentally discovered enzymes, which he named zymase, and earned a Nobel Prize in the process.
It's akin to popping the hood of a car, completely removing the engine, setting the engine on a workbench, and watching it continue to run perfectly on its own.
That's exactly what it was like. It synthesized the two opposing theories. I mean, Liebig was fundamentally correct about the mechanism. Fermentation is an enzymatic chemical process that doesn't require the overarching structure of a living organism to function.
Right.
But Pasteur was correct about the origin. Those chemical enzymes are constructed by and housed exclusively within living cells.
The sources cite Buchner's cell-free extract as a fatal blow to vitalism.
Yeah, vitalism was the prevailing philosophical belief that biological processes were driven by an unquantifiable, vital force unique to living things, like something distinct from standard chemistry and physics.
And Buchner proved you could extract the machinery.
Exactly. By proving that the metabolic machinery of life could be extracted, isolated, and studied in a test tube, Buchner basically laid the foundation for modern biochemistry.
Well, if the scientific establishment was eventually forced to accept that fermentation could happen outside a living cell, I have to imagine they were equally resistant when scientists tried to explain the other side of the metabolic coin.
Oh, you mean how aerobic respiration actually works?
Yeah.
The pushback was immense.
Yeah.
We mentioned earlier that respiration uses an electron transport chain to generate a massive 38 ATP, but we didn't discuss the exact mechanism. In 1961, a British biochemist named Peter Mitchell proposed a theory called chemiosmosis to explain it.
Can we break down the mechanics of chemiosmosis? Because if the electrons are just being passed down a chain in the membrane, how does that physical movement actually synthesize ATP?
So Mitchell proposed that it works very much like a hydroelectric dam. As electrons bounce down the transport chain, the proteins in the chain use that energy to physically pump protons, hydrogen ions, across the cell membrane.
So they're physically moving material to build up pressure.
Exactly. This creates a massive buildup of protons on one side of the membrane, just like water building up behind a dam. And these protons naturally want to flow back across the membrane to equalize the pressure.
Okay, so how do they get back in?
The only way back in is through a specialized protein complex called ATP synthase, which basically acts like a turbine. As the protons flow through, they physically spin a portion of the enzyme, And that rotational energy is used to force a phosphate onto ADP, creating ATP.
It is a mechanical, structural process, not just molecules bumping into each other in a fluid.
Yeah, and that is exactly why the biochemical establishment rejected it for over a decade. At the time, scientists were convinced that respiration must work just like the substrate-level phosphorylation we see in glycolysis.
Like a direct chemical handoff of a phosphate group.
Right. Mitchell's idea of an electrochemical gradient doing the physical work was viewed with deep skepticism, bordering on outright hostility.
He ended up retreating from the mainstream academic world, right? Moving to Cornwall and using his own personal wealth to fund a private laboratory just to continue his research.
He did. He spent years meticulously accumulating evidence to prove the consensus wrong.
And his conviction was ultimately vindicated. By the late 1970s, the experimental evidence supporting chemiosmosis was undeniable. Mitchell was awarded the Nobel Prize in Chemistry in 1978.
It really stands as a testament to the fact that scientific consensus can sometimes lag years behind mechanistically correct foundational ideas.
Okay, so we've spent this time in the abstract and the historical, mapping out electron delivery trucks and looking at the academic battles over defining them. I want to ground this in what it looks like in the physical world.
Okay, let's do it.
The sources map out some incredible downstream effects of these pathways.
The variety of end products is staggering, honestly. Consider Swiss cheese.
Oh, right, the holes.
Yeah, the characteristic holes, the eyes of the cheese, are a direct result of a specific fermentation pathway. A bacterium called Propionibacterium fortinrachii consumes lactic acid and ferments it into propionic acid, acetic acid, and carbon dioxide gas.
And as the cheese ages in a solid state, that CO2 gas cannot escape, so it slowly accumulates into bubbles, forming the holes.
Exactly.
Which actually clears up one of the big misconceptions noted in the sources, the idea that fermentation always means producing alcohol. We culturally associate the word with beer and wine, but in the case of Swiss cheese, the microbes are producing propionic acid, and in the case of dental cavities, it's lactic acid.
Yeah, the microbe Streptococcus mutans thrives on human teeth. When you consume dietary sugar, this bacterium rapidly runs lactic acid fermentation. It dumps its electrons, secretes lactic acid as its waste product, and that acid sits directly on the tooth enamel, stripping away the minerals.
So a cavity is essentially localized microscopic fermentation damage.
That's exactly what it is.
But the applications scale up far beyond food and dental hygiene. During World War I, Britain faced an existential supply chain crisis. They were basically running out of acetone, which was a non-negotiable ingredient for manufacturing cordite, the smokeless propellant used in millions of artillery shells.
Right, and prior to the war, acetone was primarily synthesized by distilling wood, but the scale of the conflict just vastly outstripped the available timber supply. Chemical synthesis routes simply could not scale up fast enough.
Enter Chaim Weizmann.
Enter Chaim Weizmann, a chemist who later became the first president of Israel. He offered a biological solution. He had patented a process using a specific bacterium, Clostridium aphetobatilicum.
But how do you take a microscopic bacterial process and scale it up to supply a global war effort? Logistically, that sounds impossible for 1915.
It required entirely new fields of industrial engineering. Weizmann's bacteria could ferment strains from corn or horse chestnuts directly into a mixture of acetone, butanol, and ethanol. They had to construct massive plant-scale fermentation vats and maintain strict sterilization protocols. Because if competing microbes infiltrated the vats, they would consume the starch and produce the wrong end products.
That makes sense.
They also had to perfectly balance the environment, because many bacteria are facultative anaerobes.
Meaning they carry the genetic blueprints for multiple pathways. They can run aerobic respiration if oxygen is present, but switch to fermentation if it's absent.
Precisely.
Yeah.
They actively sense their environment and dynamically shift their metabolic strategy to whatever is most advantageous.
Which tackles another myth from the sources that fermenting bacteria are somehow primitive or metabolically inferior. The ability to monitor the chemical environment and swap out the entire metabolic engine on the fly is highly sophisticated.
It's an evolutionary marvel, and clinical microbiologists rely on that exact sophistication today. Because different bacteria possess different sets of enzymes, they produce distinctly different fermentation byproducts.
Oh, so they use those byproducts to identify them.
Exactly. Medical labs use these metabolic profiles as chemical fingerprints to diagnose infections. The IMVC test panel is a classic example. It can distinguish E. coli from other closely related bacteria purely by analyzing whether the microbe runs a mixed acid phytonation pathway or a 2-3-butynenyl pathway.
And the clinical notes get even more intense when dealing with serious infections like gas gangrene.
Gas gangrene is terrifying. It's caused by Clostridium perfringens, which is an obligate anaerobe. As this bacterium infiltrates and ferments muscle tissue, it produces a significant amount of gas.
Creating a distinct crackling sensation under a patient's skin.
Yes, known as crepitus. And because physicians understand that this specific pathogen relies exclusively on anaerobic metabolism, they can deploy targeted antibiotics like metronidazole.
Which specifically disrupt those anaerobic pathways without harming the patient's aerobic cells.
Exactly.
And I noticed in the clinical notes regarding obligate anaerobes, oxygen isn't just unnecessary for them, it is an active threat. This completely debunks the assumption that all life inherently requires oxygen.
Oh, for sure. For an obligate anaerobe, oxygen is highly toxic. Aerobic organisms possess specific defensive enzymes, like catalase and superoxide dismutase, which neutralize the dangerous reactive byproducts of oxygen metabolism.
And obligate anaerobes just lack the genetic codes for those shields.
Right. If they are exposed to oxygen, the reactive molecules literally shred their cellular components, and they die.
Let's bring this back to human biology on a normal day. The human gut microbiome is constantly running these exact processes. When you consume dietary fiber that your own human digestive enzymes cannot break down, your gut bacteria take over.
Right. They ferment that fiber into short-chain fatty acids like butyrate, propionate, and acetate.
And those byproducts are far from just metabolic waste. They are critical signaling molecules. They provide the primary energy source for the cells lining your colon, they regulate inflammation, and they interact directly with your immune system.
Human health is deeply biologically anchored to the success of microbial fermentation.
So, to summarize the deep dive, the next time you eat yogurt or bite into sourdough bread or check a tire pressure gauge on a car running on ethanol blend, you are witnessing the output of an ancient metabolic fallback strategy. It's a microscopic engine that forced scientists to redefine biochemistry, overturned vitalism, and scaled up to arm the artillery of World War I.
It's a mechanism of survival that completely shapes the macroscopic world. But, there is one final detail from our sources worth pondering. We tend to view oxygen as the vital, life-giving default of our planet. But Earth's oxygen-rich atmosphere was actually manufactured by microbes.
Wait, really?
Yeah, roughly 2.4 billion years ago, photosynthetic cyanobacteria began utilizing a new metabolic pathway that released oxygen as a waste product.
Which initiated a great oxidation event.
Exactly. And to the vast majority of anaerobic life thriving on Earth at that time, oxygen was a lethal poison. The sudden accumulation of this reactive gas triggered a mass extinction that permanently altered the planet's atmospheric chemistry and forced evolution down entirely new paths.
That is mind-blowing. The very air you are breathing right now, the oxygen your cells desperately rely on to run their highly efficient aerobic respiration, was originally just a toxic bacterial pollutant. Think about that the next time you take a deep breath.